Okay, why don't we get going? Thank you all for joining either live. That's always a treat to have people live, or if you're catching this on a replay, we appreciate you stopping by. Let's go ahead and get started. So to recap why we're doing these, this is number 8 in our series. Understanding brain injury and treating brain injury is a gigantic mission, and we see this over and over again. And we go to conferences that are attended by thousands of people who are dedicating their lives to this study. And our goal in our reason for existence is to help along that mission, to build tools and maybe provide some education and help the smart people along in their mission. So these webinars are just another element of that. We're hoping to provide tutorials and use cases and samples and allow you to see some of the work that other people in the field are doing, how they're approaching problems in AI and machine learning and solving them, the tricks and tips and the problems they encounter along the way, give you ideas, the things that you might do in your own research. We're also kicking off some starter grants. We've got some people that we're allowing onto our cloud system and giving them computer time. If you're interested in doing that, please reach out to us. If you have feedback about the webinars, please either shoot that QR code or visit the link or shoot me an email. We always would appreciate feedback if these are useful, what could be better. Also, this is kind of a community, and so we need volunteers to speak. Ethan and I are not the smartest people in the room. He's pretty smart, but I am definitely not the smartest person in the room, and so it's much more fun and interesting to hear from one another. So we're looking for volunteers to fill out our 2024 schedule. So if you got something to present, please let us know. So this is number 8. As I said, you can go and view any of the replays at the link on the screen. They're out there for us. You can reach them on the website, or they're all on YouTube on our channel. So cool. And also for the assets, some of the webinars we're providing assets, data, and notebooks and things so you can try this on your own. You should have received an email with instructions to sign on to that site. So if you haven't, please reach out to me. We can get you added to that list and get you signed in. So it's a very cool version of our cloud platform that we've set up just for people who are enrolled in the webinars. We only have 2 more dates on the schedule. We will be filling this out. So May 17th, June 21st, mark your calendars and volunteer to speak. If you have questions, please use Zoom's Q&A feature. I think we're all pretty familiar with that now after a couple of years in the pandemic and working remotely. Today we are super excited to finally get a couple of people on that we've been wanting to have on for quite a while. Please welcome Dr. Baptiste Balança and Dr. Valentin Ghibaudo. We've known and followed Baptiste for a number of years, mostly through his work in the spreading depolarization community. And we're super excited about an opportunity to maybe work with him directly on some work that he's applying for some funding for. So it's really exciting about that. He's an assistant professor at the University of Lyon and a critical care doctor at the University Hospital, also there in Lyon. Dr. Ghibaudo is a postdoc fellow at Lyon Neuroscience Research Center. He recently completed his thesis, congratulations, in fundamental neuroscience, and he focuses on investigating connections between heart, respiratory, and brain rhythms. Today, they have a really interesting session for us. They're going to talk about an approach they use to visualize and detect features of brain injury progression. And they discuss their relationship between cortical injury and subcortical impairment. They've got some data and notebooks and visualizations. Dr. Balança texted and said, "Hey, can we break out the notebooks and show code and show stuff?" And I said, "Yes, please do. This is what we're here for." So with that, I will stop sharing and let the smart people share. So thanks, Dr. Balança and Dr. Ghibaudo for showing up, and I'll turn it over to you.
Great. I'm not able to open my video. I don't know if you want the others to see us. Otherwise, I will start sharing my screen. Okay. Share. And so, yeah, great. So, hi everyone, and thank you for attending. And during this webinar, we will show you our approach to visualize and detect some features of brain injury progression, and so share some code and some ideas that we have to visualize this interaction between brain and subcortical injuries. So let's jump directly to the point. Oh, yes, great. So why do we monitor people? So first, we can diagnose some conditions, some brain injury. We can detect new injury. We can use it to optimize cerebral perfusion, metabolism, and so on, and to look for improvement when we want to withdraw some life support, for instance. And in this lecture, we will focus on how do we detect new injuries, and mainly in the ICU, wherein we want to prevent or early detect the occurrence of secondary brain injury after primary injury. And so, as many of you do, we use multimodal neuromonitoring in unconscious patients or sedated patients that combines invasive monitoring with ICP, with PbTO2 or cerebral microdialysis, ECOG, with intraparenchymal electrodes or cortical electrodes, with non-invasive techniques such as EEG, pupillometry, or transcranial Doppler. And there are many markers of brain injury that reflects several pathophysiological mechanisms that are ongoing after those injury, either TBI, SAH, or ICH. And so there are cortical spreading depolarization that we will be talking about, some regulation of, or some brainstem regulation of the respiration or cardiovascular system, cerebral autoregulation, seizure, cerebral oxygenation, cerebral metabolism and vascularization, and so on. So we will not approach all those markers, and we will focus mainly on cortical spreading depolarization that can be recorded with ECOG, and the regulation of respiration and cardiovascular system with the brainstem. So just so that everyone speaks the same language, because I don't know if all of you are familiar with cortical spreading depolarization. So those are abnormal cortical activities that reflect the depolarization of a brain area. For instance, when you have a new infarct, like an ischemic stroke, for instance, you will have a decrease of CBF, decrease of oxygenation, decrease of glucose, a cessation of the ongoing brain activity with a flat EEG line or ECOG line. And within minutes, you will have a depolarization of every cell, neuron, and glial cells due to the lack of energy. And then there will be a complete breakdown of ions across the plasmatic membrane. And then there will be subsequent depolarization that will spread from the core of the ischemia to the surrounding areas. And this is a movie here from the team of Heidelberg in Germany, in swines, where they injured the brain here, and you can see the cerebral blood volume changes that spread across the different gyrus from the injured site to the surrounding area with those circulating wave of spreading depolarization. And when you have the wave that spread from the core of the injury to the healthy, let's say, brain tissue, you have a DC shift here that you can see on this upper curve that will trigger a complete breakdown of the ions, a cellular swelling that you might be able to see on MRI with an ADC restriction, and a cessation of the background activity that is also called a depression of the activity. So that's the spreading depolarization, and that causes a spreading depression here with a consumption of oxygen, consumption of glucose, and subsequent increase in CBF. And in between, when the depolarization crosses penumbra area or injured area, you have a large variety of reaction from a normal one to an ischemic one with, for instance, after subarachnoid hemorrhage, you can have a decrease in CBF that can lead to cortical infarcts, like delayed cortical infarcts. And from the late 90s, we can now record those events in patient with ECOG, so electrode placed on the top of the cortex, like here. And you can see in red this large DC shift here that spread from this electrode to this one, to this one, to this one. And so to be able to see those depolarization on the ECOG signal, you need to unmask what is happening below 0.5 Hertz. So you need a DC amplifier that has no high pass analog filter to be able to record what's happening between 0 and 0.5 Hertz. So this will be in our presentation, evidence in red, is DC potential changes, DC shifts. And then the rest of the usual EEG signal will be displayed in black when it's above 0.5 Hertz, so between 0.5 and let's say here 70 Hertz. And unlike what you are used to see, where you have 30 seconds of EEG recordings, here you have 1 hour recordings, so everything is really compressed. And you can see that the background activity here is really compressed, and you can see the depression of the amplitude here with a subsequent flat line. So that's the spreading depolarization that spread from one electrode to the other across the cortex of this patient. That was one of the events that we wanted to discuss with you today. And we also wanted to discuss how can we look for brainstem functions and how the brainstem centers, mainly in the medulla oblongata, regulates the respiratory rate and the heart rate vascular tone. And a way to do that we will describe later on is to look at the heart rate variability, that is the distance between two heartbeats here that can vary over time. And for respiratory rate, since most of our patients are intubated, you can have end-tidal CO2 recordings like here. So you have the expiration here, where when the patient exhales CO2, and then the inspiration phase here. And so you can measure the duration of a respiratory phase, and you can measure the variability that there is when the patient is in an assisted mode or in a mixed control and assisted mode. What's interesting when we look at some survey that might be a little bit old, 2017 now, but in neuro ICU or in neuro injured patients, almost every neurointensivist use ICP. Some use TCD, continuous EEG, PBTO2, and the more in depth you go to signal processing, such as the PRX, the less physicians are using it at the bedside. And perhaps one of the reasons for that is that it is still something that is subjective, that you need to review, that is time consuming, that requires some training and several barriers regarding how to measure it, how to compute the signal, and how to have detailed reports. So there has been many progression in how we can visualize like continuous EEG or quantified EEG or mixed EEG with other invasive or extracranial monitoring with some reports. But there is a huge need to have an automatic detection of new injuries. Could be sparing depolarization, could be subcortical injuries, but could also be metabolic disturbance and so on. And so that's what we will be talking about. So before we deploy a model that will be able to detect and provide warnings of brain injury progression, we need to train the model. But before we train the model, we need first a database. So we'll describe how we built our database very quickly. And then we need to know what we want to look for. So have manual annotations of spreading depolarization, have outcome measures, and so on. And then once we know what we want to predict, then we need to select some parameters, tune the parameters to have the right metrics to fit the model before the model can be trained and optimize the features. So we are not at the model training, but we will show you how you can select some parameters based on what you want to predict. And in our institution, we, since 2020, built a database of neurocritical care data, wherein we have multimodal neuromonitoring, like invasive and non-invasive data, combined with extra neuro data like ABP, end-tidal CO2, ECG, and so on, using the CNS monitor that many of you are familiar with, I guess. And then we combine those data with the clinical data from the IntelliSpace software that we use to give some medication and have clinical rating of the nurse and the physicians, and with imaging data. And they are patients with severe TBI, SAH, ICH, who are admitted in our neuro ICU in Lyon. And we will start shortly to collaborate with other French ICU to have a larger database in the future. And with this database, then we worked on several programming tools that Valentin will describe more in depth to analyze the data and look for those markers that we are working with.
So to say just a few words about these tools, so Baptiste spoke about this database that he made. So the first step is to manage to read this data, meaning this raw data coming from the Moberg software. So we developed with an engineer of the lab, of the CRNL lab here in Lyon. We developed a toolbox with Samuel Garcia, which we called PyCNS, which aims to first read the raw data from the Moberg. So read from raw Moberg data to Python format. And secondly, this tool aims to view the data. So this is both a reader of the raw data from Moberg and a viewer of this kind of data. And secondly, to answer to our questions about some features about brainstem activity according to the presence, for example, of spreading depolarization, we use another tool, Python tool, to detect and provide us some heart rate features and respiratory features. So this tool is called Physio because it aims to detect some physiological features. We, with Samuel Garcia, coded it into Python during my thesis, and this aims to detect the heartbeats and detect some particular timestamps, important timestamps of the respiratory signal, and that's it.
So, for instance, that's an example of what you can get from the PyCNS library. And we will show you some code afterwards. But basically, that's a visualization that you can easily have of the ECOG data. So you have bipolar montage here with the 1 minus 2, 2 minus 3, 3 minus 4, and so on. That was a 6 electrode ECOG strip. And here you have a density spectral array, a spectrogram of the derivation ECOG 3-2. And so you can again see here in red the DC or near DC slow potential changes here. And you can see a sparing depolarization occurring here and here. In black, you can have the fast activity. So that's 1 minute, 1 hour time window. And so you can easily see the depression here on the black curve. And you can see on the spectrogram here that the activity in yellow goes to blue, meaning that you have a decrease in the power of the ECOG signal. And so from these signals, you can then extract features that can further be used to automatically detect spreading depolarization. You can see here, for instance, this gray panel here that detect the duration of the depression phase. And so you can extract features like when do you have slow potential changes that spread across the cortex, and then which one are associated with the depression of the activity. And using that kind of features, there is a recent publication from Sharon Jewell in London, who worked to have an automatic detection of sparing depolarization from multimodal monitoring data. So they have that kind of software with bins of time, with PbTO2, end-dial CO2, oxygenation, CPP, ICP, and so on. And you also have an automatic detection of sparing depolarization using those features of slow potential changes spreading across electrodes and depression of ongoing activity if there was one. And you can see that for a sensitivity of 100%, that is to say that every SD detected by an investigator was also detected by the software, they have a false positive rate of 15 to 20%. So it's not here yet, but that's a first step. And that's a way to use some features to have a model that was a very simple model, but then you can train other more advanced model to automatically detect SD, and perhaps in the future have those kind of warning that either you need to review or either can trigger some medication or some intervention.
So what about some brainstem features? So as we said, we are interested in heart rate activity and respiratory activity. So the first step is to detect particular time points of the signals coming from ECG for the heart rate and from CO2 signal to detect respiratory features. So here is an example of a view proposed by the PyCNS toolbox, which aims to show the detections of the R peaks of the ECG here, for example, detected thanks to the physio toolbox. And here an example of the detections made by the physio toolbox of the respiratory cycles coming from the CO2 signal. So in green here, as Baptiste said, we have the start points, time points of inhalations, and in red of exhalations. So from these events, we can compute all the features, meaning the rates and the variability of the rates. Because here, for example, if we take advantage of the computing of the cycle duration of each respiratory cycle, for example, we can compute, we can take a time window, for example, of 2 or 3 minutes, and we can make a distribution of the cycle duration of the respiratory cycles that were detected into this time window. So here is an example of distribution of the cycle durations, but They are here computed into cycle per minute. And the idea is that on this distribution, you can see a red dot here showing the average respiratory rate. And on this distribution, there is another thing that is showed, which is the variability of the breathing rate, meaning the width of this distribution. So the idea, a good idea, could be to plot continuous time plots that will show at one time both the respiratory rate and the respiratory variability. So this is done here, for example, where you have a 2D plot with a long time window of several hours, and you have here the continuum of the red dot here, but according to the time, which is the breathing rate, so the red line. And you can see with coding by the colors, the width of the distribution, which is here a little bit narrow, and here it is much larger. So here we have an increase of respiratory rate variability. And you can do the same thing for the heart rate, thanks to the detections of the heart peaks made on the ECG signal. And you can see here the heart rate with the red line, with here, for example, a tachycardia. And you can see a larger distribution of the interbeat interval here, more here than here, meaning an increase of the heart rate variability during this time window. So this is an example of how we can see both the rate and the variability of the rate of the respiration and of the heart in just one plot, one figure.
And so what we are interested in our lab is this interaction that we can have between cortical abnormal activities such as pairing depolarization. So with the same, again, using the PyCNS library with the physio library, you can have an easy display of here a spreading depolarization that occur on the electrodes 3 and 2, and that's spread here. And you can investigate the relation or the time relation between this abnormal activity, this spreading depolarization, and the changes that you have in brainstem regulation, or at least in a surrogate of brainstem activity, which is the variability of the respiratory rate and the variability of the heart rate. And this is an example that we are intending to publish in Lessons to Learn in Neurocritical Care about how we can evidence brain death in patients. And that's an example of a patient with a poor grade subarachnoid hemorrhage with an intracranial hemorrhage, which was operated. You can see here the bone flap. There was ECOG electrode placed on the cortical surface. And due to the clipping of the aneurysm and the acute rise in intracranial pressure, the patient developed an ischemic stroke in the right hemisphere with, you can see here, injury of the mesencephalus with right and then left mydriasis. And so over the first days in the ICU, she evolved to brain death on day 6. And you can see that on day 3, there was several repeated spreading depolarization. And then here on day 6, a while after there was this mesencephalus injury, there was a complete cessation of ECOG activity with a first sparing depolarization. And then if we unmask the real DC potential, you might be able to see a large negative infrastroke potential reflecting permanent cortical damage. And so when we plot the same display as the one we evidence or we show you a few slides earlier, you can see here, so that's a zoom on this part here when the patient underwent brain death. You can see here the sparing depolarization that spread from one electrode to the other with a permanent cessation of brain activity. And here you have another depolarization. And if we had on this graph unmasked the real DC potential, you might be able to see a large DC shift here, but it will go out of the limits of the plot. So we just use a near DC red signal. And you can see that 15 minutes later, there is a permanent cessation of respiratory rate variability. That is to say, the patient was in only a control mode without any assisted respiratory cycles. That could also be evidenced with an apnea test, but that's an easy way to see that there was a cessation of spontaneous respiratory cycles. And you can see that within minutes, there was also a decrease of heart rate variability with the tachycardia reflecting this Cushing syndrome that we can observe after brain death. That's also a nice way to evidence a permanent or at least a cessation of the respiratory trigger from the brainstem. And so that you know that now you can be able to make an apnea test and eventually do other tests such as angiographic CT to confirm brain death. So now I will give back the voice to Valentin, who will describe and present some of the codes that are behind those nice plots that we described here.
So yeah, I already spoke about these two toolboxes that are available on GitHub. So PyCNS to use its function of reader of Moberg data and viewer, which we can merge with the physio toolbox to detect the brainstem activities as we showed. And I propose a notebook at this, which is available with some codes, a sample of codes at this link. And so I'm going to switch to a notebook, which is in this GitHub link, to give some examples of how we do it. So here is a notebook I prepared with different parts. So the first part is to import some, for example, some toolbox. So here are some classical tools of Python to store some data. Here I import, for example, the physio toolbox. So we made here in Lyon. Here I just import some path for going to my data. Here I import some functions from the PyCNS tool, meaning the reader function and the viewer function. And here are just some tools I coded to give rise to the views and to the figures we showed during our presentation. So the first step here is just, for example, to select the ID of the patient, meaning the patient folder where the data are stored. And here I just show some meta information, some metadata. So these informations come from a table that come from Baptiste. So these are from our own data. So this was a woman which lasted for 6 days in the intensive care unit. Here, I just load some other metadata which come from Baptiste, but that are included in the Moberg data because he manually detected the spreading depolarization. So here I just show the content of the table of the patient, and there are several lines with, for example, some event name. So here, for example, a spreading depolarization with its date of occurring. So this will allow me to jump in some time windows to show these events manually detected by Baptiste. So let's go into the opening of the Moberg data, which is quite simple. So here I prepare the paths that go to the patient data, and I give this path to the reader function of the PyCNS tool. So here the data are pre-loaded, and it tells me that there are several streams of data here. And for our concern, we can see here some CO2 data for the respiration, arterial blood pressure, EEG, ECG, etc. So I can open, for example, the data that are inside this ECG stream in order to detect the ECG peaks of the patient. So here I open the ECG stream, and I'm going to load the data thanks to the function get data of the PyCNS. So I'm going to load the data from this ECG stream, and here I select the time window when I want to open this data. So here I just want all the data of the patient. So I select just none to none, and I get this raw ECG, and then I can give this raw ECG to the compute ECG function of the physio toolbox in order to detect the R peaks. And when they are detected, I get a table here, which is the detections of the R peaks. So I have many detections, and for each R peak, I have the time point and the date of this R peak. The same thing can be done with the CO2 signal to get the respiratory cycle detections. And so here it is running, but it is fast, almost fast. And I get this kind of table, meaning a table with many rows, and each row is a respiratory cycle with its own features, meaning some durations, cycle frequency, inhalation duration, exhalation duration, etc. And I have for each of these time point a time after the beginning and a date. And from these detections, the first thing is, I think, to see if these are true detections. So here I take advantage of the viewer function of the PyCNS tool to see the quality of my detections. So I give the whole reader of the patient, and I give some external plots, which I prepared, meaning some external views aimed to show the detections of the ECG that I made and of the respiratory cycles. And here we can set just, for example, 30 seconds. And here I see the ECG of my patient with the date, and I can jump from inside the signal and see my detections and jump by 6 hours, for example. And I see that here I have truly detected the R peaks of the ECG and the respiratory cycles. So, okay, my features, my time points are well detected, so I can see things like the plots we showed during our presentation. So here I take advantage of the viewer function again of the PyCNS tool, and I provide to the viewer some external views that I prepared, meaning a view that allows to see the spreading depolarization, for example, a view to see the density spectral array of a derivation here, for example, a view to see the respiratory rate and the heart rate. This can be parametered to see more or less things. So here I just set, for example, 2 hours of signal. And this will show me the kind of things we showed during our presentation. So here, for example, I jump to the 10th row of the Baptiste table, meaning I jump to spreading depolarization, and I see in this time window 2 spreading depolarization here and here, with the depression of the fast activities that can be seen of the density spectral array. And I can see that during this time window, the breathing rate is stable, meaning that the respiration is fully controlled. And I can see the heart rate, which is quite stable too, with a low heart rate variability. I jump, for example, to another spreading depolarization window. So this is done, and we see here a spreading depolarization, with after that an increase of heart rate of respiratory rate variability and of heart rate variability. And just a lasting example, here I jump to the brain death that we showed during the presentation, and we see this loss of respiratory rate variability after the second spreading depolarization of the window with the Cushing reflex here. So that's it. Here is a notebook with some Python code, and all these things are all these things are available on the GitHub link I provided. So at this link, there is the notebook, which is here, and some the tools I spoke about. So we return to the presentation, and it's time to conclude.
Yeah, I think we are on time to conclude. So we described how we try to identify features of brain injury, and we focused on sparing depolarization and brainstem functions, but you can do the same with other features. We think that there is a high need for automated detection of those events at the bedside, because this will be a way to have a broader use of those tools in neuro ICU by neurointensivist and nurses. And we just shared with you some of our tools with the PyCNS, and perhaps in the future it could be implemented in clinical care software such as the Connect one developed by Moberg Analytics. And perhaps there will also be some AI tools in the near future. So we are happy now to take any question that you may have, and you can have our emails here if you need to send us some questions or remarks. Thanks for your attention. Thank you.
Well, thank you guys so much. This was, I have to say, when we first envisioned, or when I, first was thinking about how we wanted, what we wanted our webinars to be, it was exactly this. I mean, queuing up a really interesting problem from a clinical perspective, getting in. Ethan and I have been chatting in the background, diving into the code and looking at all your GitHub repos and your packages and stuff like that. This is amazing and so cool. I can't thank you enough for coming on and sharing. While we wait, if anybody has any questions, waiting for those to get queued up. So I mean, you mentioned, you gave us a little teaser at the end there about AI. What direction are you taking this in now? Are you looking at starting to do predictions? It seems that's the next step, or what's next for this?
Well, I think that the next step for us will be to have automatic detection some events that we want then to try to implement at the bedside, because I think it will be hard to have a prediction of the long-term outcome with short snapshots of multimodal monitoring for a few days. But I think that the first step that as a neurointensivist I would need is to have something that helped me at the bedside to have an early detection without spending hours of coding and reviewing the data. So perhaps our first step will be to automatically detect some adverse events, such as spreading depolarization, but could also be other things.
Cool. I'm sure you're familiar with the work that Dr. Hardings and Srikar are doing out at UC on that, right?
Sure, that was something that we discussed before, and I'm looking forward to try what we implemented in the Connect software and how it works at the bedside.
Yeah. Cool. Any more questions? Let me see if there's anything get queued up here.
I can hop in. Thank you very much for presenting. It was very interesting. I haven't thought to look at that data in that way, so it definitely kept me interested when you were talking through the presentation. The plots are beautiful. Have you thought about extending this analysis to correlate it with some of the other features you mentioned? You kind of alluded to that in the beginning with cerebral autoregulation, PRX. And then also, do you think that you could extend what you're doing to look at other correlative measures between modalities beyond what they do for PRX?
Well, that's a great question. Most of what we are doing is going back and forth from the time domain to the frequency domain, and to visualize the variability in the time domain and so on. And so we are starting to work on the interaction that there might be between those variabilities, cerebral signals, and cerebral autoregulation. We submitted a poster to the neurocritical care for neurotrauma meeting that will be in Cambridge next September, I guess, about that. But that's something that we are looking into, and so perhaps we could share some ideas once we have made some progress on that side.
Thank you. And I know Craig asked about whether the next step would be for prediction. Have you gotten any evidence that prediction of these CSDs are possible?
Well, there are some publications, and I think that Judd Hartings have something going on with Python based software to automatically detect spreading depolarization. On our side, we started to try some basic detection. We are not here yet, but I mean, if other people already have done the job, perhaps we will not spend much effort in having something that already exists.
Cool. Thank you very much.
Cool. Any more questions? Well, having none then, thank you guys again so much for coming on and sharing this work. It's so cool. And I know Ethan and I will be diving into the rest of your code.
Sure.
It's really really a great discussion. We really appreciate you coming on and sharing this cool work with us.
Well, that was great talking with you and with the audience. Don't hesitate to send us an email if you have questions that comes up afterwards.
And to use the codes that we presented, if you want.
We definitely will. Cool. All right. Well, thank you guys so much. With that, I'll wrap it up and close out the call, and we'll talk to everyone and see everyone next month. Thanks for showing up or catching us on the replay, and we'll talk with you soon. Thanks.
Thank you.
Thank you all.
Bye.